Thank you so much, Professor Siddiqui. And I would like to say thank you for those who have been responsible for bringing me here. And I just feel incredibly privileged and honored to be able to be here on this visit and to have this opportunity to talk with you this afternoon. I'm going to begin with my first slide. And this is something we think about. When we look at a venerable individual, such as the person pictured here, it raises the question, how do we age? And I'm not thinking particularly of, can we live forever or something like that. I'm thinking, what's the quality of life? What's the health of this person as such an individual ages? What goes on during aging that makes somebody's life as they age a good life or a more difficult life, perhaps accompanied by lack of health? Now, aging is a multifaceted process and an organism as complex as a human being. And so in order to study it, one really has to study it from a variety of different ways. And I'm going to be telling you about just one of the many aspects and facets of aging. And the way the science of telomeres and telomerase that I'll tell you more about in a moment has taken us into this question has been to show us an interesting connection with what happens in aging, and that is the facet of aging characterized by increased susceptibility to diseases as one ages. And I've just listed three very common kinds of aging-related diseases, not all of them by any means: cancer, heart disease, diabetes, for example. Another aspect of this question that we as biologists want to understand is factors influencing aging, and particularly this aspect of aging, the increased susceptibility to the diseases that characterize aging, an increase in frequency with age, is how much of the influence on these processes is non-genetic, we could call them something like environment, life factors, non-genetic, and how much is due to our genetic makeup. And so I'm going to be touching on those themes as I talk about today's topic, which will be aging, and particularly the aspect of aging. And I want to show you somebody who's successfully aged here. This is Dr. Marguerite Vogt of the Salk Institute, the now late Marguerite Vogt, but until her into her 90s, she could keep doing what she loved doing most, and that was come to the lab and work. And here she is in the lab working at a tissue culture hood here, doing what she loved most. So this is how we would like to see aging, not living to be 500 years long, but being able to do what we love to do for as long as possible before one dies. And I think she's an example of successful aging. And so how do we look at aging? Well, if we looked at some of the 100 million cells in her body, let's say, you'd have a cell and you would have the nucleus inside the cell. The heart of the cell is the nucleus, and inside the nucleus, if we then looked under a microscope, we would see the genetic information. And so these blue objects here are the chromosomes, very long DNA molecules, all compacted up and folded up, as Professor Siddiqui mentioned. And there are several of these chromosomes. And this cell is about to divide. And you can see each of these chromosomes is actually double because the DNA has completely replicated itself. And so what will happen when this cell divides to become two cells is that one copy of each chromosome will go to the two daughter cells. Highlighted in red are the ends of the chromosomes, the telomeres. And that's the aspect of chromosomes that we're studying. And I'm going to tell you now about the very basic science of that. And then I want to move into how this has taken us into the question that I framed, which is, how do we age and what things might influence it and what mechanisms might be at work? Now, the genetic information is carried in the DNA of the chromosomes, our entire genetic blueprint, and that information is protected by telomeres and telomerase, and I'm going to describe those now. Telomeres are the bodies that cap the ends of chromosomes, shown highlighted again in this pinkish red color. An analogy. It's always good to have an analogy or a metaphor. Now, if you've got a very expensive pair of shoes, unlike the ones that I would get, for example, right? What you'd get for your money for buying this expensive pair of shoes would be a shoelace. And the shoelace we can think of as being rather like the chromosome, the long DNA. And the shoelace, though, for this expensive pair of shoes, is capped off by something that even has a special word in the English dictionary, an aglet, which caps off the end of the shoelace. And so we can think of the telomeres as being the aglets of our genome. The telomeric DNA, as I'll show you, has binding sites for protective proteins, and these nucleate a higher order complicated architectural structure at the ends of the chromosomes. But the bottom line is it keeps the ends from fraying. My PhD student generously donated his sneaker shoelace for the purpose of illustrating that this is what happens if you don't have a good tip to your shoelace, that the ends can fray away. And that's a fairly good analogy for what happens to the ends of the chromosomal DNA. This fraying is clearly adverse. So now we'll home in on a slightly more molecular view of things. So now if we think, here's a chromosome. Now if we zoomed in on the very end portion, we would find that the end of the chromosomal DNA was made up of very short repeated sequence motifs of building blocks of DNA, and they don't code for any genes. But what they do is that they make a kind of an architecture of DNA which acts as a landing pad or a scaffold that attracts a whole suite, a whole group of protective proteins that bind the DNA. And the DNA, as you remember, is double-stranded DNA, and there's even a single-strand portion that protrudes from the end, and special chromosomal proteins bind these two portions of the telomere. That's what the telomere consists of, the underlying repeated DNA sequence motifs and the protective group of proteins. Okay, if that array of repeats and the protective proteins, if for some reason it wears down and becomes too short, that sends a warning to cells. This is an unsafe situation because this end is in danger of losing its protective capping ability. The cell then elicits in the cell all sorts of warnings and alarm systems because the cell wants to fix this situation. And it will stop multiplying, for example, because all its DNA can't be properly replicated if it's fraying at the ends. And so the cells, if the cells should normally be multiplying, can no longer keep multiplying. The fraying ends can become sticky and stick to other parts of the DNA, making the chromosomes now irregular in structure and leading to genomic instability. So a telomere that has become too short is a danger to a cell. And sometimes cells will actually kill themselves rather than continue with this very unstable situation. Telomere shortening happens naturally in the course of things. That's the problem. It happens. And why does it happen? There's some technical reasons to do with how DNA replicates. Remember I said most of the DNA all has to replicate every time a cell divides, but if the DNA machinery can't replicate the very end, which turns out to be, for complicated biochemical reasons, the case, then the telomere, each time the chromosome replicates, every time a cell divides, then the prediction was it would get shorter and shorter and shorter. And this was actually predicted simply knowing the enzymology of DNA replication back in the 1970s. And so the prediction was that perhaps eventually this would limit, in some as then quite unknown way, it would limit the ability of cells to replicate. And in fact, Olovnikov, on purely theoretical grounds, not knowing any of the molecular details of this, predicted perhaps eventually you might lose some sort of genetic information and perhaps that would lead to senescence, a prescient idea as it turned out. So what does telomerase do then? I've just told you what the problem is, that there's this wearing down just in the natural course of things because of the way DNA replication works. And so telomerase turns out to solve the problem. And so I just want to tell you how we found this. And we were using an experimental system, a single-celled pond dweller called Tetrahymena thermophila. This is a scanning electron micrograph of this single-celled organism. And the reason we chose this was that it happened to have a very large number of very short chromosomes in it. And so that means a lot of ends, right? If you have a lot of very short linear chromosomes and you've got lots of ends, it was a good experimental system in which I found what the telomeric DNA sequence looked like. And it was also a good source, it turned out, for an enzyme that would act on telomeric DNA and, in fact, make it. And so the enzyme is called telomerase, and it's the solution to this wearing down problem. Because now, if we now zoom in on the very end of chromosomal DNA and now show the building blocks of the DNA, so here's the double-stranded DNA, here's the protruding single-strand, individual nucleotide building blocks shown here, just in a very simplified form. What telomerase does is that it's an enzyme made of protein portions, of which one is the core, and then there's another portion which is an RNA component built into the enzyme. And within that is a short DNA or RNA sequence, since this is an RNA shown in blue, which acts as a template for synthesis of nucleotides which are synthesized onto the end of this DNA, thereby elongating the DNA. And this can happen over and over again. And so this can solve the problem of attrition from the ends. Other enzymes come in and fill in the gap down here. So what good is that? We discovered this enzyme, Carol Greider and I discovered this enzyme back in the 1980s in tetrahymena. What's the point of this? So in the course of examining the enzymology of tetrahymena, we found out something quite useful. So as I said, we have tetrahymena thermophil and we found telomerase in this organism. Now, this is an organism which actually is, if you will, immortal. So long as you feed it enough food, it will keep multiplying forever. So it must have good repair mechanisms to keep its DNA in good enough shape. But essentially, the cells just keep dividing forever. There's no apparent limit to how much they can keep multiplying. And they have, as I said, plenty of telomerase. Okay, plenty of telomerase, the cells are immortal. And then genetically, we used a trick to inactivate the telomerase in the cell. And what happened was that now, as the cells went through repeated cell divisions, gradually the telomeres got shorter and shorter and shorter and shorter and shorter. Progressive shortening of telomeres was seen then, and the cells, when they got to a certain point of shortness, the cells ceased to divide. So they became from immortal, with lots of their natural telomerase, to, when we inactivated telomerase, they gradually ran the telomeres down just as predicted and became mortal. Okay, so that said, telomerase was not a luxury for cells. They needed it. So without it, it's like you stuck an arrow into the heart of the cell. You killed the cell. Okay, so telomerase then is maintaining the ends of chromosomes. It can elongate the chromosome ends. And what it does then is the chromosome ends get elongated, but of course they're actually still subject to the shortening processes that I told you about. It's just that telomerase will balance out that shortening. So balancing out the shortening, there'll be lengthening and shortening, and so there's a balance kept. So overall, the overall length is maintained, and the cells can then keep on dividing. So that's the principle of how it works. So what about in human cells? Okay. A lot of work has been done, and I can summarize it as saying telomerase in human cells can be a good thing or a bad thing, depending very much on the context. In other words, on what cell type one finds the telomerase. In the context of cancer cells, telomerase is distinctly bad news. Okay? Now, a cancer cell is not a normal cell. It's a cell in which a number of genetic and epigenetic changes have taken place. This is a rogue cell where many changes have taken place. What telomerase is doing, at least in the most simple form, and I'll tell you in a lecture tomorrow, a more specialized lecture about some other things it's doing, but the basic thing it's doing is it's simply keeping the telomeres replenished, just as I told you it can do. So the cancer cells have lots of telomerase, and they can keep the telomeres replenished. And so these rogue cells that have gone wrong for so many other reasons and are deaf to all entreaties by the body to not keep multiplying inappropriately, these rogue cells are given, if you will, the green light by the excess telomerase, and they can keep on multiplying. Telomerase itself did not really make them cancerous. Many other changes did. Okay, and in fact, because they rely so much on the telomerase, it's a very interesting target to think about molecularly as a target for anti-cancer treatments, and there's various activities of ours and other labs to try to look into that possibility. That's all I'll say about telomerase in cancer cells. Now I want to talk about telomerase in normal cells, and now it's a very different picture, because in the context of normal healthy cells of the body, cells that are not cancerous cells, telomerase, as I'm going to show you, is rather good news. Okay, So let's just imagine the various situations that could hold in a cell. Now, I told you about the situation in an immortal cell such as tetrahymena, where if you have plenty of telomerase, there's a balance is kept between addition, that is addition of DNA by telomerase, and shortening, the natural shortening, attrition of telomeres that happens in the course of rounds of DNA replications as cell divides, and other nucleases, which are enzymes that can actually chew away on the ends of chromosomes. So telomerase can balance out those shortening influences, and so the cells could keep dividing. So for a cell to keep on multiplying, there are certain ones that we'd like to keep multiplying, for example, the germline cells, or effectively certain stem cells whose undiminished ability to keep on replicating is important for organism and from generation to generation survival. Okay, so now what if we knock telomerase down? Now we're going to disrupt that complex, as I told you, because the telomeres will get too short to protect the ends. So, they're going to become prone to fraying. And now the situation could be a bit different. So I told you what happens when there's no telomerase. Now what if there's some telomerase? What will happen is that the shortening is happening, but now it's out of balance. And then even though telomerase might be adding, eventually the race is lost. Eventually senescence occurs. And then you can imagine, of course, another shade of gray in which now you have, say, less telomerase. And so it'll now happen faster. So you can see the point at which the telomeres will get too short will depend a lot on the balance between these processes. And we know a great many genetic players into what affects the balance. But what we're finding, as I'll show you, rather to our surprise, is just the amount of telomerase seems to be a rather important major determinant of that. Okay, so now let's consider what happens in the many decades that we all hope we will have in the lifespan of a human. Okay, so now the time frame is becoming very, very long. So let's go back to our question. This person has been clearly alive for many decades, and what about these aspects of aging, or this particular one, the increased susceptibility to disease? And as I mentioned before, what about this question of how much of this facet of aging, that is the increased susceptibility to diseases, would be genetic versus environmental? Now I'm going to be asking that question in the context of telomere maintenance. So a generalization is when you could read sort of any review in aging or gerontology journals or journals that deal with the science of aging, and you would see something like this. So I've just chosen this example. A generalization is that elderly people who demonstrate exceptional longevity have generally been spared the major age-related diseases, which I mentioned were common examples: cardiovascular disease, diabetes, and cancer, which diseases are responsible for most deaths in the elderly. So long as conditions are good in terms of sanitation and people are not dying early of infectious diseases, for example, then if they can live long enough, then they will be subject to these sorts of diseases. Exceptional longevity, and I won't go through the information for this, but that has a very strong genetic component. In other words, good genes can really help you live a long time, but you have to get through all this, first of all, in order to get to the point where you can have enough longevity to see if your genes are going to do you any good, to summarize a lot of information. So these are of great interest then, since the elderly who have got to the point of being elderly are going to have to be concerned with these sources of mortality. Okay, so what do we learn about telomerase and telomeres? And as usual, the human clinic and human genetics are incredibly informative for diseases that can be very rare, but they can be very informative, even though rare. And so one such example was a disease of insufficient telomerase. So what happens in the now some dozens of families where this has been found is that on the chromosome that bears the gene for telomerase RNA, that RNA component of the telomerase enzyme, what happens is that if there's a mutation, then in these rare inherited disease situations, people die young, they don't make it to old age, A primary cause is progressive bone marrow failure. They simply run out of bone stem cells, it seems. And all it takes is one of the two copies of the telomerase RNA gene to be mutated in one form or another. Okay, so one form from one parent is fine, but there's been an accidental mutation in the other gene. And there's a variety of these in different families that have now been seen. But what they do, in effect, is they knock out the function of the telomerase RNA. So in effect, the person is running all their cells on half gene dosage of telomerase RNA. And the consequence, as I said, is usually progressive bone marrow failure. People do not make it beyond middle age, and often it's well before that. They have shorter telomeres. So that tells you telomerase in humans, at least down in the range of getting down from 100% gene dosage to 50%, that's very limiting because the telomeres are much shorter than in the general population. Their immune system becomes exhausted because, of course, their bone marrow, which is the source of their immune system cells, is running out of stem cells, it turns out. Interestingly, such individuals, it's now clear, are cancer prone. And that relates to this aspect of telomeres capping the chromosome ends and preserving from genomic instability. So that is actually not an unexpected thing. And I want to point out that the cancer proneness is all about the fact that telomerase and telomere maintenance is important in the healthy cells, right? Now, we don't have enough telomerase in this situation. And so that's not allowing the healthy cells to be maintaining their stable genetic information. The ends are becoming uncapped, they're getting very sticky and causing genomic instability. And it's thought that that is likely to be the reason why these individuals are also cancer prone. Okay, so this is certainly a characteristic disease of normal aging, but this is a rare disease. But it told you a very important genetic fact, and that is that if you want a full lifespan to well beyond what would be considered middle age, then both telomerase RNA genes have to be functioning properly, and so the quantity of telomerase does matter. So that's something you could only find out from human genetics because the causality is very clear. Okay, now what about the rest of humanity, people who don't have this rare genetic disease? So what happens in vivo in terms of common variations in telomere maintenance in human beings? If you look at telomere lengths in people, you'll see that there's quite a wide variety of telomere lengths in people. In a very general way, the telomeres tend to become shorter as you get older, but there's actually a very large degree of variety and scatter in that relationship. So can one see anything informative? And something that in effect was like a prospective study, if you will, was done by Richard Coffin and colleagues and published back in 2003. And what they found was that they were looking at fairly elderly people, not the exceptional longevity people, just fairly old people. And they found that people who had shorter blood cell telomeres in the mid-1980s, and the blood samples had been frozen so they could come back and look at the telomeres, they, in the subsequent 17 years, had higher mortality rates. In fact, from all causes, their survival over the next 17 years was actually worse if they had shorter telomeres than the people in this group, this cohort who had longer telomeres. Interestingly, that included higher mortality from one of the most common diseases of aging, heart disease, and also from infectious disease, which is interesting given what I just told you about the haploinsufficiency, in which telomerase insufficiency clearly impacts on the immune system. And in fact, these individuals with the haploinsufficiency of telomerase RNA usually do die from infectious diseases. They can't fight off infections. So interestingly, that was an interesting observation. It didn't tell you what caused what. It simply said there is an association. Okay? So we got into this question in a very different way, in a collaboration with a colleague of mine at the University of California, San Francisco, Dr. Alyssa Eppel, in the Department of Psychiatry, in which she was interested in a very non-genetic aspect of life, and that's chronic psychological stress of a kind which has been well defined by people who study this as being clinically problematic. And I'm going to point out here, since the word stress is a rather broadly used word, that the kind of stress I'm talking about here is the kind that really has known ill effects on people. Now, there's sort of the good stress, the stress, you're going to run a marathon. I'm sure I'm under the good stress now, giving a talk in front of this large, distinguished audience here. So this is called a challenge stress, though, and actually that doesn't hurt you at all. So it's the kind of stress where the person feels the situation is out of their control, they have an unpredictable nature to it, and they don't feel they have the resources to cope with the situation. And so it's a situation that many life situations can be incurred by many life situations. So we can think of it as a kind of an input signal into somebody's physiology. And many studies have associated this with a variety of diseases, including diseases that are diseases of aging, such as heart disease and diabetes, for example. So this has been seen in clinical studies. Okay. So my colleague, Elissa Eppel, and several other colleagues in studies that she has organized, we joined together, and Jerlyn in my lab and a couple of others joined in with this as well, and decided to have a look at what happened to telomere maintenance in some very well-designed studies that this group of people were designing with respect to chronic stress. So I'll just tell you what the general finding was, and then I'll show you a bit more. And that was chronic stress. So I've sort of shown that as a brain, because that's where that's where that input is coming into the brain, the chronic stress, if you will, through no doubt immensely complex human physiology. We're seeing there's an impact on telomere maintenance, and the chronic stress is making the telomere maintenance worse, and so the telomeres get shorter, as I'm going to show you. So one study was with some mothers who are the biological mother of a child who has a chronic illness, and for whom the mother is the primary caregiver. And in the United States, that's commonly a fairly unsupported situation where the mother is often sort of not able to get a lot of help for this situation. So it's up to her. And so it has all the characteristics of the kind of stress I said has been known to have clinical outcomes. It has the unpredictable nature, the out of control, often inability to identify resources to cope with it. So it was chosen as this objective stressor situation. And in fact, there was a control group of mothers as well. So this is very carefully designed by my colleagues to try to take out as many confounding factors as possible. So it was a very carefully designed study. And so the two quantitative questions asked that I'll deal with first was the level of perceived stress, which is a self-report, actually, but it has turned out to be quite a good, reliable measure in many studies of how a person is coping with the stress, and the duration of the caregiving, and this is now a number of years, so it's a very clear number, nothing to do with perception of the stress, but just the number of years. Were either of these perceptions, this can be reduced to a quantitative number, where the worse the perceived stress, the higher the score, and of course the number of years is obvious. Were they related to markers of cell aging? And the two in particular were telomerase activity, which I've shown you is important to have enough to maintain telomeres, at least as the genetic experiment showed us, and telomere length. And we use the cell type that healthy people will give you, and that is white blood cells, a blood draw. And so basically, if you divided people so all the studies we do, of course, are completely blinded samples, and we analyze all the samples blinded, and then they go back to the study, and then the code is broken, and then you look and see what happens. And so what happened was that people who were under high stress had less telomerase than those under low stress by this perceived stress measure. And indeed, the telomere length was noticeably shorter, and this, you can see the confidence levels, they really didn't overlap. The telomeres of the high-stress people were generally shorter than those under low stress. Okay. Another way of showing the data would be as this table here. So the perceived stress was related negatively to telomere length. In other words, the worse the perceived stress, the shorter were their telomeres right across the scale for both the control group and the caregiver mother's group, and the lower was their telomerase activity, somewhat weaker relationship. Now, also the years of caregiving was, and obviously that applies just to the caregiver group, were also comparably related. And that was an interesting thing because, I'll just show you a bit of the data here. So here was the telomere length, and this was the number of years of caregiving. And so then one goes through all sorts of tests to see whether there were confounding factors. Could it just be explained by age or body mass index and lots of things like this? And essentially one couldn't make these relationships go away. And so we were forced to conclude that the more years that the mother had been in this objective stressor situation, the lower, after correcting for everything we could think of, the lower were her telomerase levels and the PBMC, that's the white blood cells, telomere length. So very simple numbers here. We're just treating these as quantitative parameters here. The cells are a mixture of cells. In new studies, we now divide them up into subcell types. But it's a very interesting thing that just taking this rather crude, very simple number, we started to see these sorts of relationships. Because of the relationship with the chronicity, the time or duration of the objective stressor, that thing coming in, if you will, from the outside or from the brain, we think we can put an arrowhead here, and we think that it's reasonable at this stage to conclude that this is causing this to happen, that the excess shortening and lower telomerase is related to this parameter here. So when you do one study like this, then of course you immediately worry about, is there something peculiar about the study? And you want to replicate the study. And so this has now been done in a variety of studies that we have partly completed. Some are still ongoing. Some are still, happening in the future. But we are now seeing this same kind of thing happening in other completely different cohorts. So it was not an accident here. So just to repeat, the first finding was that the more years she'd been in the situation, the worse was her telomere maintenance, as judged by the lower telomerase and the shorter than expected telomeres. And so what we have is then a sort of relationship that we could think of. Oops, well, I was going to animate something. But okay, so let's go on to another study. So this is now another situation of women who are, again, in a very difficult caregiving situation. Now they're postmenopausal, as opposed to this premenopausal group I just described. They have a husband or a spouse or a partner who has dementia, and they're the primary caregiver. It's often Alzheimer's disease, but it's sometimes other kinds of dementia. And again, we saw the same relationship. I'm just showing you one part of the data where the telomere length was related to the perceived stress. And again, when we try and correct for every other parameter, including age, we can't make this kind of relationship go away. Okay, here's another very interesting kind of a stressor situation. This is for the postmenopausal women group. And an interesting thing is it's called cognitive dietary restraint. And essentially, it's can be quantified on what's called the Dutch Eating Behaviors Questionnaire scoring, but it's essentially a kind of behavior in which, it's really a disorder in some ways, in which people will try to diet, but they don't succeed. And in fact, they usually put on weight, if anything. It's not caloric restriction, by the way, which is very different and can probably have beneficial effects, as shown in animal studies. But this is called cognitive dietary restraint, sort of repeated failed attempts to go on some sort of dieting. And anyway, again, we saw this relationship here with the worse was this on the scale here, the lower was the telomere length. So again, the same directionality, not implying any causality here, of course, but the association was clear. And we could, again, try to correct, as I said, for all other sorts of possible mediating or confounding factors. And I've just shown three here, and you can see this relationship. The telomere length with restrained eating, that was the relationship that held out. And we also saw it with the premenopausal group as well. And this is the postmenopausal group I just showed you the data for. So we're seeing the same pattern again in different groups of women. This is another rather interesting one. This is called dispositional pessimism, and it's a scale in which the person answers quite insightful questionnaire, and so then their degree of pessimism, which is a relatively stable trait of individuals, is simply plotted against telomere length. And again, it was rather striking finding. Okay, so we can sort of simplify this and say, well, there are sort of life factors, things outside the genome, which are affecting cell aging in these ways. And so I've just shown you some of these few examples that we've seen here. So the relationship would be somewhat like this. We know, if you will, signal input in the brain. So we know about this. But now, then there's going to be very complicated physiology, which you could sort of think of as signal integrating and processing. And there's going to be all sorts of, feedback loops and interactions. It's going to be immensely complex. And I have no doubt that the physiology of the body has all sorts of complex mechanisms here. But we can treat it in this rather simple way and then say, well, it does have an effect on telomere maintenance. And so now the question is so does that matter? Okay, does it have any impact on something measurable like disease? So let me tell you what we found. In the initial group, we found that cardiovascular disease risk factors were interestingly related. So just to put this in perspective, for example, a large study published just a few years ago, which involved something like 29,000 people in 52 countries at 6 of the continents, so covering humanity pretty broadly. The risk factors, the prominent risk factors for cardiovascular disease are, no surprise, smoking, poor lipid profile, high blood pressure, diabetes, abdominal obesity, and psychological stress. Now, that's the same kind of chronic stress that we were talking about, which I just showed you in this study, this well-controlled study, and there's a couple of studies now that I've mentioned, was related to lower telomerase activity. So we had one of those that I told you about. But actually, this study measured a lot of things and, in fact, measured all of these. Not diabetes, because everybody was carefully chosen to be healthy, to be in this study, but in fact, resting levels of insulin and glucose, which if high, are a predisposing factor to diabetes. Okay, so all of these measures were intact. So what did we find? Some interesting relationships. Let me just give you a couple examples. I always like this one because, if ever you need to have more messages not to smoke, I like this because look at the non-smokers. They show the whole range of telomerase activity in their blood cells. This is, and look at the smokers right down here. Okay, so not good. And here's an interesting one. The heart function of these individuals was really quite distinct depending on their telomerase. Now remember, everything is blinded. So when these measures of their heart function in a particular lab stressor situation I'll tell you about was measured, of course, there was no clue about the telomerase, but when the data were put together, here's what emerged. So first of all, the individuals sit and they're resting for a while. And so then you can measure their heart rate and their pulse pressure, which of course, as you know, to be good, should be low. And so the people who have the low telomerase, the half of people with low telomerase had higher heart rates and higher blood pressure than the other half of the cohort who had the high telomerase. So the high was lower in a good way. This is high frequency heart rate variability. It's a matter of adaptability of the heart. So when it's high, it's good. So the high telomerase people had the higher value. So that was good. Okay, so now my colleagues do a very nasty thing to these individuals. Okay, so they're in this lab situation, very controlled environment. And what they do is they ask them to anticipate giving a videotaped speech and then give the speech and then to do some very unpleasant mathematics. And the reason it's rendered unpleasant, this whole experience, is because trained personnel sit in the room stony-faced, no expression, as they proceed through these things here. So they get no reaction from trained personnel who are looking at them all the time, not reacting. And you can imagine this is not comfortable. And what happens is that people's heart pulses start to race. And so what happens, though, is rather distinct between the two groups. So the heart rate goes up much more in the low telomerase half of people than in the high telomerase. Their pulse pressure shoots up, and then their high frequency heart rate variability, it actually is no better. I mean, it should be higher rather than lower. So very distinct, almost really two different groups of people based on this really simple median split, just splitting the telomerase levels into two halves, the high and the low half. And the pattern you see here is well known in cardiovascular clinical research. This sort of overreactivity and poor heart rate variability, this poor adaptability, that is known to be associated with risk for cardiovascular disease. So in other words, this is not a good pattern in the low telomerase half of this cohort. Interestingly, I just showed you a couple of examples. All 6 of the risk factors, well, not diabetes, because as I said, this is a healthy group, but one predisposing factor, fasting glucose. All of these 6 risk factors, or a surrogate in the case of fasting glucose, were all associated with lower telomerase activity. Didn't really show up much as telomere length, but lower telomerase activity was the one that went with all 6. So in fact, lower telomerase activity was associated with the known risk factors, the major risk factors for cardiovascular disease. So we're seeing a link then in people. Essentially, we can say from these sort of data, low telomerase alone, even without telomeres having got a lot shorter, is associated, no causality implied, with 6 major risk factors for cardiovascular disease. Now, does this matter? Well, it turns out it does. And here's a very recently published study where one can look at the rate of telomere loss. And this is quite an interesting study. This was a healthy, high-functioning elderly people, 70 to 79. And in men, what was done in the study was to measure telomere length at a 0 time point, and then 2.5 years later. And these individuals were followed out for 12 years. And what happened was that those whose telomeres in this time interval at this first 2.5 years, those whose telomeres were maintained, this is their survival curve. Those whose telomeres shortened, and there was they had to shorten quite a lot, by 15% or more, that was their survival curve. And in fact, the chance that you would die of cardiovascular disease was 3-fold worse if your telomeres shortened. So this was a sort of anticipation, if you will. If there was a rate of shortening that was negative, in other words, if telomeres shortened, here, then over the succeeding 12 years, then the rate of cardiovascular death was higher. So it seems as if this association is holding up. And this kind of study then is one of the very early ones going on, where now rate of telomere loss, not just simply looking at telomere telomere length at one time point, but looking over time is important. And remember, rate of telomere length loss, remember that's going to be related to various things, but we'd predict it would be related to level of telomerase, because the less telomerase you have, the less able the cells will be to stave off telomere shortening over time. Okay, now, people haven't looked at telomerase much, but they've looked in now lots of cohorts at telomere length, almost always in cross-sectional studies. What I just showed you was a recent exception, but in fact, a lot of diseases of aging have been linked to shorter telomeres, and I will just list a subset of the many studies out there just to highlight common diseases of aging in which short telomeres, generally in blood cells, which is where one can get the most convenient access to cells to examine telomere status, has really been seen over and over again. So in cancer, cardiovascular disease, vascular dementia, certain degenerative conditions associated with aging, such as osteoarthritis and osteoporosis, diabetes. Certain risk factors are also tracking with telomere shortness, and pulmonary fibrosis, a relatively common and quite deadly condition, which does get worse with aging, also have been linked with telomere shortness. So putting it all together, where do we've seen all these associations, what do we think is going on? So I'll try and tell you what we do know and what is known and what is pieced together but is not proven. Okay, so first of all, as I said, genetics has been very, very helpful. And of course, there have been mouse genetic studies done, although mice only live a couple years and humans can live up to 100 or so years. So the timeframes may be different, but the results are reasonably convergent in certain ways, and that is that if you make telomerase deficient in mice or humans, telomerase going down does indeed have, as a consequence, telomere length maintenance is worse. Telomeres definitely get shorter. So we know for sure there's causality here. Okay, and there's a lot of science, laboratory-based and organism-based science, which says that if the telomere length maintenance is compromised, as I told you, when cells sense their telomeres are no longer protecting their genetic information, they will set off a series of alarm signals and the cells will cease dividing. And so this will reduce the ability of cells to replenish. So if it's a tissue that has to replenish throughout life, such as the immune system, this could be serious. And we saw in case of haploinsufficiency of telomerase, the genetic disease in humans, that indeed that was serious. And indeed, there is disease impact of various kinds seen in this known causality situation. Okay, now another thing that's been well established in the clinical world is that indeed chronic stress is something which does also have disease impact. And that's been seen for a variety of kinds of diseases and immune function, for example. Okay, so now the question is, what about telomerase? Are these related? So, okay, what do we know? Well, as I said, we can make a case from the duration of the caregiving situation that this is causing lower telomerase and therefore less ability to maintain telomeres, because we saw shorter telomeres and lower telomerase in people who'd been subject to more and more years of chronic stress and who were subject to various kinds of chronic stressing situations as well. So we think we're fairly confident about that in humans. Okay, and as I said, a lot of science in the laboratory and with model organisms and so on. We're pretty confident about this arrow too. Okay, so we're on good ground here. Now, this is the part that I would say is unproven. Is this known inability or diminishment of the cell renewal capability, which we know can be caused by this? You should say, we know good based that can be caused by this. But is that actually the thing that's having impact on disease that would explain why chronic stress, for example, has a disease impact? Or is it completely independent? Or most likely, as is the way of biology, it'll probably be elements of both this and this. But we could certainly make some very plausible, well-backed up arguments here, and we can make some reasonable arguments about this argument. So that's what we do know and what we don't know. Yes, so everybody wants to know, what can we do about it? So we can't do anything about our genes right now. So we get that. For the foreseeable future, what can we do about the non-genetic impacts on telomere maintenance, which, as I've argued, we have reason to think may be having this disease impact, and where we've seen these associations with higher mortality rates from the diseases of aging. So there are a variety of studies in progress now to prospectively look in randomized groups of people, randomized to different things such as stress interventions, to try and see what can be done. So I've told you all about the bad end of things, right? When things go from normal to worse. What happens if things go from, say, normal to better, or from worse to better? Can there be any reversal of this? And as I said, it hasn't been looked at in enough prospective and randomized trials to say for sure. But interesting indication comes from a study which was simply cross-sectionally looking at telomere length. It looked at a very large group of twins, and interestingly, the twin of the pair who exercised had longer telomeres than the twin who didn't exercise over a year period. So this was a cross-sectional, just looking at a snapshot of the telomeres, but there was this relationship that exercise was associated with longer telomeres in the twin who exercised. And so looking at twins was valuable because it then equalized for a lot of genetic input. And we know there is genetic input into telomere length maintenance, as I've described in the extreme case of the genetic mutations, but there are common variants that are starting to show up as having influence on telomere maintenance in humans. And so we predict from all the science we know that this will have a good effect on the ability of cells to keep on self-replenishing. So I think the science of this is probably fairly certain. And so, of course, the question is this, exercise, is it because telomerase got improved, or why did the telomere length maintenance get apparently better in the twin who had exercise? So we don't know the answer to that, but that seems like a very reasonable hypothesis that that's happening. And that would be something provable in prospective, as I said, randomized controlled study. So back to the main question. Genetics can account for exceptional longevity. That does seem to have a big genetic component to it. But the very common diseases, such as cardiovascular disease and diabetes and cancer, which account for most diseases in elderly people, most deaths in the elderly, those ones, I hope I've shown you, there's also some component that we can clearly start to discern is having a non-genetic component. Well, that's certainly been well established, but the new thing is that we think that this is being associated with impaired telomere maintenance. Okay, so to summarize what I've been saying, forms of chronic psychological stress are associated with low telomerase and telomere shortening. Does this matter? We think that there's reason to think that it certainly is at least a very interesting association because low telomerase and shortening of telomeres in human cells in vivo is associated with disease susceptibility and shorter life, and as I described to you, there's some reason to think perhaps also may contribute to disease susceptibility and shorter life. Okay, so I'll end up with a picture. So in a sense, what have we learned here? Well, poor telomere upkeep, both from genetic causes and through non-genetic influences, is associated with, as I said, perhaps contributes to, susceptibility to cancer and other diseases of aging and shorter life. And so this is another kind of a picture. Sorry to mix my picture metaphors for telomeres, but you sort of think of a telomere as this beautiful structure like a tree at the end of the chromosome, and we don't want it to look like this. Thank you, and I will just put up the slide that acknowledges over the many years I've been doing this research that I've told you an update about today. These bodies and institutions have been responsible for letting us do the kind of research that we are able to do and that I was able to tell you about today. Thank you.